The human cell is a marvel of molecular organization, with countless proteins working in harmony to sustain life. But what happens when these proteins become damaged or are no longer needed? Enter the work of Irwin Rose, a biochemist whose discoveries revolutionized our understanding of how cells maintain order by disposing of unwanted proteins. Born in 1926, Irwin Rose devoted his scientific career to solving one of biology's fundamental puzzles: how cells selectively mark and destroy specific proteins. In the early 1980s, while working at the Fox Chase Cancer Center in Philadelphia, Rose collaborated with two Israeli scientists, Ciechanover and Avram Hershko, on what would become a groundbreaking discovery. Together, they unraveled the mystery of the ubiquitin proteasome system, the cell's sophisticated garbage disposal mechanism. This system operates through a process called ubiquitination, where proteins destined for destruction are tagged with multiple copies of a small protein called ubiquitin. The beauty of this system lies in its precision. The cell can specifically target individual proteins for degradation while leaving others untouched. This selectivity is crucial for regulating countless cellular processes, from cell division to immune response. Rose and his colleagues discovered that this tagging process requires the coordinated action of 3 different enzymes, working in sequence like a molecular assembly line. Once a protein is tagged with ubiquitin, it's recognized by a large protein complex called the proteasome, which unfolds and chops the tagged protein into small peptides. The significance of this discovery extended far beyond basic cell biology. Disruptions in the ubiquitin system have been implicated in numerous diseases, including certain cancers, neurodegenerative disorders like Alzheimer's and Parkinson's, and inflammatory conditions. For their pioneering work, Rose, Ciechanover, and Hershko were awarded the Nobel Prize in Chemistry in 2004. The Nobel Committee recognized how their discovery had transformed our understanding of cell function and opened new avenues for developing treatments for diseases linked to protein degradation problems. Throughout his career, Rose embodied the spirit of scientific curiosity. He was known for his methodical approach to research and his willingness to pursue fundamental questions about cellular machinery. Even after his major discoveries, he continued working well into his later years, driven by an insatiable curiosity about the molecular mechanisms of life. Irwin Rose passed away in 2015 at the age of 88, leaving behind a legacy that continues to influence modern biology and medicine. Today, drugs targeting the ubiquitin proteasome system are used to treat certain cancers, with many more in development for various diseases. The story of Irwin Rose reminds us that understanding the most basic cellular processes can lead to profound medical breakthroughs. His work decoding the cell's protein disposal machinery has given us not just deeper knowledge of how cells function, but also powerful new tools to combat disease at the molecular level.